Don't worry, this is not going to be a striptease. No one wants to see that, but I was asked to undress, as it doesn't happen often. You already heard about places where the sun never shines, dark holes, in both talks, and I will be talking about something different. I'll be talking about how we can trick the immune system to treat cancer. So let's begin. When I'm saying immune system, I'm talking about one specific kind of immune cell that's called macrophage. And that's really just a fancy word for a pretty much ubiquitous type of cell. It's all over your body that is very good at eating up stuff that could become dangerous to you. This stuff could be bacteria, viruses, cellular debris, so basically just trash, or foreign substances like microplastics or asbestos. And macrophages are excellent at eating up those things before they can really become dangerous to you. But we haven't even talked about the scary stuff yet, and actually had a perfect foundation from the previous talk about those spontaneous mutations that arise in your body all the time. Here's a little shocker for you. In the last 12 minutes, while you were listening to the last two excellent presentations, almost everyone in this room will have had at least one cancer cell show up in your body. Let me repeat that. While you were enjoying your time here, listening to some excellent talks, almost everyone in this room had at least one cancer cell mutate in your body. And you know what? That's fine. It happens all the time throughout your whole life, and your immune system has it under control. Your immune system is extremely good at finding and eliminating those rogue cells before they can cause any problems. Only when the cancer cells can team up on your immune cells and can suppress immunity can the cancer multiply, spread throughout the body, thereby endangering and often ultimately killing the patient. So my research focuses on how we can arm the immune cells, more specifically on how we can arm macrophages to treat cancer and keep up this great defensive wall against those random mutations or against an established cancer. The problem is it's very hard to modify, to engineer fully committed immune cells. But it's very easy to modify stem cells. And so what I've been doing over the last 3 years is to find a more effective way of making as many macrophages as possible from stem cells. But of course, the macrophages I make in lab don't really look like a yellow smiley face. That's just a metaphor. They look like this. And then the stem cells I use to make that don't really look like green circles on a blue backboard. They look like this. The mutation I use is called 14G2A, and it's a surface receptor. It's on the surface of the cells that directs these cells against the most common and also most dangerous, most lethal type of brain cancer called glioblastoma. And what I've found out is that when I start from this new stem cell type that I was a part of discovering, the so-called turn cells, I can produce about 10-fold more of these macrophages and of other immune cells per culture volume and per input cell than has previously been possible with conventional methods. This is a huge thing for accessibility because, as you can see here, right now donation-derived cells are by far the cheapest option, but they are very hard to bioengineer. And all cell line or stem cell-derived cell types are easy to bioengineer, but about 1 order of magnitude more expensive. So this 10-fold gain in efficiency could really make this among the cheapest options, if not even the cheapest option, while also being very amenable to bioengineering. So what can these cells actually do? Well, they go towards sites of inflammation, so towards parties, if I may use the metaphor from the previous talk. And as you can see in the bar graphs on the right, they do this way more efficiently than conventional stem cell-derived cells. When they arrive at the site of inflammation, so for example at a site of infection or at a tumor site, they can eat up what they find there. In this case, they have eaten up green glowing bacteria, and they can destroy what they have eaten up by producing reactive oxygen species, breaking down those partying cells, so the cancer cells or the bacteria. And as you can see on the right again, they do this way more efficiently than conventional stem cell-derived immune cells, and they also do it at a better viability. So they live longer, meaning that you probably don't have to repeat the therapy quite as often. And lastly, quite recently, I think this result is from last week, I have succeeded in placing this immune receptor on the surface of the cells, so they now have the GD2, which I will now start testing these cells against various cancer types, including glioblastoma. In summary, I have created a way more efficient way of outfitting immune cells, arming immune cells against cancer, and I will now start testing it against various solid cancer types, including breast cancer, ovarian cancer, and brain cancer. And with that, thank you so much for your attention and for listening to me rant about the immune system and about cancer. And thanks to my working group and to my funding sources, without which I couldn't have done this research.